Okay, lecture 10, we'll continue our discussion on DNA sequencing analysis. So during the lecture 9, we talked about the principles of Fred Sanger's sequencing methods, which was based on the dideoxynucleotides, how they can be incorporated into the DNA when it's replicated, and then how they can terminate the replication whenever they are added. So through the random termination of the replication, all those replicated products will be sorted out based on the sizes. That's how the Fred Sanger's DNA sequencing works, right? But like other technologies, that technology has been advanced through invention and innovation. So the sequencing method that Sanger developed actually was changed through the technological advancement. So these days we don't use that old-fashioned Sanger's dideoxynucleotide sequencing anymore. So we have modified. Okay, actually it's more modified now. The Sanger sequencing is not used anymore, but we'll talk about the advanced form of Sanger dideoxynucleotide sequencing method. The difference is, yes, we still use those deoxynucleotide, but we don't do the replication in 4 separate test tubes. Instead, what we are doing is we are doing replication in all one single tube. Okay, so you're going to add, template DNA and then a primer, okay, in a single tube, and then 4 normal DNA nucleotide and DNA polymerase, of course, okay, and then the dideoxynucleotide, 4 of them are all added together within a single tube. Okay, then how is it going to work? It can still work because you're adding enough template DNA, enough DNA nucleotide, and then enough dideoxynucleotide, but each dideoxynucleotide is labeled with some kind of fluorescent signal generating molecule. So for example here, dideoxy-A nucleotide is labeled with green fluorescent molecule, dideoxy-G is added with yellow fluorescent molecule, dideoxy-C with blue, dideoxy-T was added with red. So 4 different fluorescent signals can differentiate what dideoxynucleotide is added at the end of the replicated product. So replication is happening in a single test tube, okay, under the PCR condition. So whenever each dideoxynucleotide is added, that will stop the replication. Now it has a T at the end, another T at the end, but this one has G at the end, C at the end, right? So they also have the fluorescent signal, okay? So that signal can be picked up if you have a sensor that can detect the fluorescence. So when this mixture is separated on an electrophoresis gel, instead of using a slab gel, we these days use a small capillary gel. That's very thin gel, but its resolution is good. So when these nucleotides are sorted out on a thin gel, yes, they will be sorted out based on their sizes, and then when they are running through the gel, there's a sensor that can pick up the fluorescent signal. Okay, depends on what nucleotide is added. That fluorescent signal will be picked up at the end, and then the machine can read out, okay, so this one has maybe T at the end, next one has T, next one is G. Continuously it can read all those sequences. Okay, so that's the advancement. So we call this as a cyclic sequencing method. Okay, so because you save time because you don't do 4 replication reaction. Instead, you can do it in 1, and you use fluorescent labels that labels all 4 dideoxynucleotide, and you are using very sensitive fluorescent detection technology, and the reading will be done by the computer. So at the end, you'll get the results like this, fluoroprogram. So the machine will pick out which dideoxynucleotide was just passed through the sensor, and it'll read out the sequence of the nucleotide. So if you go to this website, okay, copy again and paste that website address, okay, and you can see that animation. Okay, so that's called cyclic sequencing. Okay, so the bottom line is how it works. Again, it's going to use that DNA replication process: template DNA, polymerase, nucleotide, normal nucleotide, DNA primer. Okay, but you're going to do that in a single test tube. And now you're going to add 4 different dideoxynucleotides with different fluorophore signal added at the end. You're going to add all those 4 into a single tube, and then you're going to do the replication. Okay, again, denaturation at the high temperature, and then cool down so the primer can bind. Okay, once cools down, the primer binds, and then polymerase comes and grab the tail. It starts the replication, right? So when it does that, it's going to add whatever the nucleotide it sees on the template DNA. Opposite complementary nucleotide will be added there. Okay, but there's a chance, normal DNA nucleotide could be added, then it continues. But if it adds dideoxynucleotides, then it's going to stop. So right there, dideoxynucleotide A was added because there was T there, then it terminates the replication. Okay, no more extension there after that, right? But you're not going to do it just once. There will be multiple template DNA, multiple nucleotide, multiple polymerase, so you can do a lot of these. They will all have different termination with different dideoxynucleotide at the end. So you have a mixture of all those in a single tube. Okay, then what do? Okay, you sort them out through electrophoresis, capillary gel electrophoresis. What you're going to do? Transfer the sample to gel, and then you're going to sort them out based on the sizes, right? Okay, electrophoresis is being done, and the machine has a very nice sensor that can pick up fluorescent signals. Okay, so it can read what fluorescent signal is running through the gel. There you go. The sensor is there. The nucleotide is running through because electrophoresis is happening, smallest to the largest. It picks up what nucleotide it just passed that site. Okay, it reads the dideoxynucleotide. Okay, that's the sequence of the newly made strand, right? So it's called electroporogram. Okay. So that's a sequence of the newly made strand. So all those replicated products are sorted out based on their sizes, from the smallest to the largest. And their tail, what deoxynucleotides were found, were detected, okay, by the detection technology, and they are read out. Okay, that's the cyclic sequencing, more efficient way of doing the Sanger dideoxynucleotide sequencing method. Okay. So dideoxynucleotide-based sequencing is a very efficient one. That's been the main sequencing method that's used for about 30-40 years. Okay, when I was a graduate student, that was the sequencing method I used. I spent a lot of time on it, so I got used to it. But dideoxynucleotide sequencing had some pros and cons. There's another sequencing method which is not very popular because it's using a lot of toxic chemicals. But if the dideoxynucleotide sequencing did not work because of some issues, there are some issues and conditions where dideoxynucleotide sequencing would not work. I'm going to talk about that. Then this is the alternative, I guess, DNA sequencing method. Again, this is a kind of old technology, may not be used anymore. So it's called chemical sequencing method or Maxam-Gilbert sequencing method. Okay, there's a big difference in the principles of the sequencing method between deoxynucleotide and this chemical sequencing method. We'll talk about that and see how they are different. Okay, so this sequencing method that's based on the chemicals is developed by two scientists, Maxam and Gilbert. Okay, so the Sanger method is of course developed by Fred Sanger, and this is developed by these two gentlemen. Okay, so the way it works, this sequencing method does not rely on DNA replication process. That's a big difference. But dideoxynucleotide sequencing is relying on the DNA replication process and then a random termination based on the dideoxynucleotide, right? But this one is not that way. Instead, the sequencing method depends on the chemicals that can modify your DNA bases, and then chemicals that can also cut up the modified DNA bases. Okay, so examples are like these chemicals, like hydrazine or dimethyl sulfate, which is DMS. and then formic acid. Okay, and these are kind of a very strong mutagen because they modify the structure of DNA bases. Very strong mutagens. Okay, and then another chemical is like piperidine. Okay, so these chemicals can also cleave the phosphodiester bonds. Okay, so by combining these kind of chemicals, you can modify bases and cut up those DNA molecules that are modified. So let me show you this diagram before that. So let's show you. So DMS, this chemical, it's a very nasty chemical that can methylate the base structure of G, okay, guanine base, and then formic acid can also modify the structure of A and G bases. Okay, so DMS and formic acid will target G or A and G. Okay, and this chemical hydrogen can modify T, thymine and cytosine, T and C bases, and hydrogen itself can also act on the C bases, okay, with the presence of salt. Okay, so these chemicals have very specific base they are targeting. So DMS, G, formic acid, A and G, hydrogen, T and C, with the presence of salt, hydrogen targets C. Okay, so if you combine these strong mutagens with the presence of the other chemical, that piperidine, so that's what you're going to see. You're going to modify some bases, and once the bases are modified, you can cut the nucleotide that are modified. Okay, so the way it's going to work is, so you will have to have a bunch of DNA molecules. Okay, and that DNA molecule, so that template DNA, although it's not going to be used as a template for replication, you will just have to have the DNA that should be sequenced out. You will have that in large quantity, and then that DNA will be exposed to those chemicals like DMS or formic acid or hydrogen with the presence of strong salt. Okay, and then after the chemical treatment is done, they will be exposed to piperidine, and piperidine will cut up those modified bases. And once that's done, you're going to separate those DNA molecules that are modified and cut on a gel, okay, to identify which part of the DNA was modified and cut. Okay, so that's how the sequencing would work. So in other words, it's kind of this way. So there's enough amount of DNA molecule that's labeled with something, so because you can visualize them using a gel electrophoresis. So that's maybe end label with maybe radioactive isotope or chemiluminescence or something that will give you a signal, right? And then that DNA will be modified and cut using those strong chemicals. Okay, it may be modified and cut on G basis or A and G basis or C and T basis and C. Okay, so their cut sites will be different. Okay, and these fragments will be sorted out on a sequencing gel, DNA electrophoresis gel. Then you can read out the result, right? So it depends on which fragments are cut showing up, and so from this test tube, you're going to see all the G sites will be cut, right? And this result, you can see all the A's and these G sites are cut. This one will show you all the C's and T sites are cut, and this one will show you all the C sites are cut. By comparing the result from all those 4 tubes, okay, giving you different DNA cut pattern, you can identify where are the G nucleotide that's located within this DNA, because G will be cut here and there, okay, but if it's cut here and there, and then there are more additional cuts that are not found here, those are going to be A's, right? Same goes here, between here and here, you will see the C cuts, right? But the cuts that are not showing up here, but it's showing up there, those will be A cuts, okay? So by looking at the cut pattern, you can identify the sequence, nucleotide sequence of your DNA. That's how this method works, okay? So, and then why is this method used at all? Because Sanger method seems to be more straightforward. It's based on the replication. You'll get more DNA made, okay? But chemical method, it's kind of nasty because you have to deal with those strong mutagenic chemicals, and then it's not going to replicate. You will have to have a bunch of DNA at the beginning. So that's not easy. But why do you have to use this method? And there's a reason for that, because there's a condition where Sanger dideoxynucleotide method would not work efficiently. Okay, so we'll talk about that, where it's not going to work efficiently.